Good morning, everyone. We're going to go ahead and get started now. So thank you all for coming here this morning. It's a beautiful July morning for this. I'm Maureen Sarter. I'm director of the Omics and Bioinformatics Core for the M-LEEaD Center here at Michigan. If you're not familiar with M-LEEaD, it is an NIEHS-funded center that aims to study life stage environmental exposures and disease. And so we have hosted 4 summer omics learning seminars this summer, with this being the 4th of those seminars. And so this morning, I am very pleased to welcome our speaker, Dr. Bhramar Mukherjee. She is collegiate professor and chair of biostatistics. She's also professor of epidemiology and global public health, and she has several other roles. She's a faculty member of MIDAS. She's co-director of the Cohort Development for the Precision Health Initiative here at Michigan, and she's also associate director for cancer control and population sciences for the Rogel Cancer Center here. As I was preparing for this, I was wondering how she has time for all of these different roles, and in fact, that's what we were just chatting about. Her research involves studying statistical associations and developing statistical methods to study associations among genetics, environmental exposures, and cancer. And today she's going to talk about the Michigan Genomics Initiative and how more recently she's been studying analyses with electronic health records. And she'll be talking about some of the challenges and opportunities presented by working with this data. So let's give a very warm welcome to Dr. Bhramar Mukherjee.
Thank you very much, Maureen. Big data, omics, electronic health records, these words have become such cultural touchstones in today's biomedical research. So it's a pleasure to be spending an hour talking about some of my own queries and exploration of this world and this world of buzzwords, and also see if some meaningful science comes out of this buzzworld. So I'm going to talk about Michigan Genomics Initiative. This is an institutional initiative, and since 2012, and the way I designed my talk is actually in 3 parts. But before I begin my talk, I have to acknowledge the contribution of several people without whom these slides will not be possible. So Lars Fritzsche at the back, so he has been a really cornerstone for the Precision Health Initiative at Michigan. And then Max and Lauren are in my lab, and I plagiarized like shamelessly from Matt Z's slides, so I'm very thankful to them. So the way I designed this talk is actually the part 1 is talking about infrastructure, that what can Michigan Genomics Initiative give, and what kind of data, and what is the vision for creating this integrated digital data ecosystem for the institution, so that many of you in the room who probably are not geneticists, but maybe doing environmental health or social sciences, how different people from different prisms and lenses can use this data resource. So the first part is going to be about the resource. The second part is going to be my own exploration in trying to use this resource for some work in genetics with my lab. And so I'll introduce this concept of phenome-wide association studies. We are very familiar with genome-wide association studies, but when we have access to electronic health records data, we have access to the medical phenome, and we can study a spectrum of diseases simultaneously. So that gives the concept of phenome-wide association studies. And then in part 3, this will be my conscience as a statistician and epidemiologist talking. I'm very tempted to use this data because this is large, this is very exciting, but at the same time, it can produce some dangerous weapons for math destruction. So how are we going to sort of combine that and sort of deal with the statistical issues? And I'd like to argue that there's tremendous information in this data, but you have to be really, really careful, because electronic health record data was not designed for research. And so when we're trying to use this for population-based research or biomedical research, there are issues with sampling, misclassification, selection bias that you have to acknowledge. knowledge, and you have to be very careful. But that also presents some tremendous opportunities for research, methodological research in survey sampling, in statistics, in biostatistics, and epidemiology of how to use these non-probabilistic samples where we do not know why an individual is there in the data set. How do you use that for novel methodological work? So these will be the 3 points that I'll try to drive home today for you, and I hope that everyone in the audience will see something that they resonate with in each of these 3 parts. And please feel free to stop me at any point of time, because this is supposed to be a conversation, not a monologue. So the Precision Medicine Initiative, so in 2015, Barack Obama announced this, and in his State of Union address, that seems so far. And so this was, and the Precision Medicine Initiative of the White House on a national landscape was actually about collecting personalized information. So I want to underscore that collecting granular level of personalized information to keep ourselves and our families healthier. And cancer and diabetes was actually posed as 2 sample diseases from which the preliminary work is going to be done, and then that is going to be mimicked across a spectrum of diseases. And what do you mean by granular level of information? So if we actually collect large ensembles of data sets with large number of variables, then probably we can find individuals who are very similar, not just in terms of genetic architecture, but in terms of their social exposure, in terms of their other covariates, and find prediction for someone who is walking into your clinic as we ensemble these other large data resources. So that's sort of the holy grail of precision medicine. From what we know, can we predict the future? So that's the crystal ball that we are all looking for. So as soon as this announcement this initiative was launched, Francis Collins and Harold Varmus, the then director of NCI, wrote this beautiful piece in New England Journal of Medicine, which emphasizes that as we are creating these large-scale biological databases, including genome sequences and other forms of omics data, then it is very important to develop computational tools for analyzing these sources of data. So the importance of methods which will harness the tremendous amount of knowledge in this data and convert this to actually knowledge and actionable knowledge. You need tremendous task force, a computational task force, a domain science task force in order to pull this off. So that is why the movement in Michigan has been involving so many people from so many schools, so many departments, because you do need all kinds of expertise in order to make sense of this data and to utilize it in a meaningful, compelling way. So as reflecting on the Precision Medicine Initiative 4 years ago now coming back to precision health, and I'm going to talk about some of the other national initiatives that are going on, not just national, international efforts that are going on in a moment, but the Precision Health Initiative at University of Michigan, that's what most of this talk is based on. It's a cross-campus initiative, and it's funded by the Provost's office, and several schools contributed to this, public health, medicine, and engineering being very, very much a leader in this space. And the idea is that can we create an integrated data frame where people can use from different perspectives and angles for agnostic discoveries as well as lookup, so that it can serve as an accelerator and incubator of scientific ideas, generating scientific ideas and reproducing and replicating scientific ideas. And so the hope is that several investigators, like for generations to come, is going to be able to use this data resource for their research. So Michigan Genomics Initiative started in 2012. And the brilliant thing, and I'm going to talk about, who were the cheerleaders and champions of this study. So what is brilliant about this study is actually a consent form, a one-page consent form, which actually consents to linkage the clinical health data to biospecimen samples for research purposes, as well as any kind of data that could be linked through identifiers. And this also allows people, for our researchers to recontact patients. So this is a very progressive and bold kind of opt-in consent form, which has really been effective in terms of recruiting and building a large inventory of participants in the Michigan Genomics initiatives. And I'm going to tell you about the sampling frame and where these patients are recruited in a moment. But initially, the cohort started as a part of like pain research, and it started in perioperative setting, and it was mainly recruited by Chad Bhramar from the anesthesiology department. So. Stewards of the data, as I said, that it takes a village, probably a country, in order to make sense and harness this massive amounts of data, processing, storage, acquisition, translation, visualization. So there is a lot of people working, and there are some people who are on these slides, and I apologize to many who are not. And I could have a slideshow presentation only on those faces. So, there are stewards of the clinical data, of the genetics data, and also there are ensemble in terms of how the data is being integrated and curated for use by researchers across campus. So the genetic data is, so when people consent, there is a blood draw, and then the blood is archived in the central biorepository, and the sequencing core genotypes it, and there is a team of, and actually an army of people in biostatistics and human genetics. They all work on the data and develop quality control algorithms so that this data is usable, including genotype imputation. So I'm not going to talk about the details of quality control of the genotype data. And then there is a clinical data, and these clinical data, and I would like to sort of iterate that these electronic health records are actually a digitized version of a patient's medical chart. So these are all the information that are being recorded when you go for a doctor visit. And so this is mostly administrative data. That's what we have to recognize, that these are diagnoses, procedures, lab results, prescriptions, are primarily used for billing purposes. And the EHR, and it's sort of health system specific. We still do not know that how to really link across Michigan, for example. If I am going to St. Joe's tomorrow, then how to link and connect my EHR with the Michigan EHR. We have not found out an amicable way to do that yet. And there is a lot of advances going on in terms of patient-mediated donation of their own EHR in that space, so that we have a more holistic view of what is going on with an individual's healthcare. So it's a historical record of health and treatment. And again, it's not primarily research-oriented, but they can be queried and used. So that's sort of the main point of this talk, that how can we have principled ways of looking at this data and dissecting this data. So of course, MGI is big data, and with big data comes big challenges. So we have many, as you can imagine, that with 60,000 patients, there are numerous medical visits, medical encounters, and after imputation, a large number, more than 20 million genetic variants per sample. I'll show you some recruitment data, 65,000 consented samples, and then our analysis cohort would be approximately 40,000 people whose genotype data and the EHR data has been linked. But the data is getting bigger, because genetics and electronic health records, though this has information in terms of your disease diagnosis, in terms of labs, prescriptions, procedures, it does not have everything. So we are making a conscious effort to collect data on behavior, lifestyle, environmental data, social data, family history data. So there is a sub-study of MGI, which is actually this MyPact study, where we also have on a subset of people high-density mobile health data in terms of Apple Watches, continuous stream data on steps, on other markers of cardiovascular health. So this is a tremendous potential for different people to write grants, write papers, and so I really want to share that perspective, that how we embarked on this journey. So I want to speak about the infrastructure a little bit more, as was mentioned, that I'm one of the co-directors of the Precision Health Cohort Development Working Group. And it's my real pleasure to work with two brilliant women, Amy Cohen from Engineering and Linda Elizabeth from School of Public Health, on this developing the MGI cohort. And that has two facets. That what additional type of data are we going to put into MGI so that it's usable by a large number of investigators, as well as how to expand and make the cohort more diverse. So I'm going to say that this has been a steady growth in terms of enrollment, and the numbers are going to vary from slide to slide because we are recruiting patients every day, but approximately 65,000 will be the right thing to say. So initially, the bulk of the cohort is still perioperative, where it started, but then there has been other cohorts, because investigators in several expertise and specialty clinics have actually taken advantage of MGI and have started recruiting patients from their clinic. So as you can imagine that the recruitment, the patient profiles in different clinics are going to be quite different, perioperative, and then we have MEND, which is the endocrinology, diabetes and endocrinology patients, and then we have mental health outpatient patients. So this is sort of the composition of the cohort, but when we put them together, as a statistician or epidemiologist or a scientist sitting in the audience, you think, how do I put this together? Because the sampling and the patient profiles of these three different clinics are going to be quite different. So how are we so there are tons and tons of interesting statistical issues. And we're also trying to partner with, to enrich the diversity of the cohort, because our catchment has been primarily Caucasian white population. So what are we thinking? And so what are the sum of the cohorts? And I just wanted to share this plot over time, how steadily MGI recruitment has actually gone in a fairly consistent and stable and linear manner. And so these are the time frames when the other clinics started recruiting. So that's why they are not the large proportion of the cohort, which is the diabetes and endocrinology cohort, mental health cohort. This is the Apple Watch cohort. And the latest one is a pediatric cohort called Big Bird. So what is happening to this process, right? Let us take a step back and think about this. Originally, it started with a one sampling mechanism, but then investigators are interested in getting, have their own research questions and collecting the data. And the idea is that everybody will be able to use this data, which is coming from different sort of perspectives, and merge together. This is probably going to be the model, that there will be a healthy sort of a cohort coming from perioperative, but then there will be subsamples and subcohorts, which is based on individual investigator interests. So MGI, as compared to biobanks around the world, I wanted to give you a little snapshot of that. This paper, I really am very proud of my postdoc, Lauren Beasley, for putting together this incredible paper, and Max as well, which actually is a review paper of all the biobanks around the world, and it's getting outdated by the minute. So I'm sure that by the time it's published, this landscape is going to change. But these are some of the cohorts around the world. So biobanking and ensembling people with electronic health record data and genetics is not specific to Michigan. Today, we are going to talk about two particular studies here. And as you can see, that the landscape is across the world. There are initiatives in China, Japan, Europe, all over the world. So we are particularly going to talk about UK Biobank and Michigan Genomics Initiative in most of our work, because those are the data sets that we have used for our analysis. So one thing I want to mention for researchers here is that UK Biobank data, with 500,000 people with different domains of data available for research to all, has been a tremendous resource for progressing science in a very rapid pace, where everybody can double check their analysis and can have access to the data, including genetics data, imaging phenotypes, which are very hard to collect on a large number of people. So US, in terms of creating this national cohort, I think US is a little bit behind, but also wants to go larger, right? So million people with a lot, so it's all of us. So this is the cohort. And also the NIH's All of Us has a very special initiative in terms of enhancing the diversity and oversampling underrepresented minorities. So that in that perspective, when the All of Us data comes out, it will be another resource for researchers all over the world to look into lifestyle, environment, and biology together. So this framework of we have been siloed in science about studying one factor at a time, just the environment or just the genes or just the lifestyles or just the social perspective. I think this movement of getting everything on an individual gives you that idea of precision medicine. And I'd like to say that precision medicine, I like to use the term precision health because there is a lot goes on before treatment. And in terms of lifestyle and social perspective, precision health is probably a more compelling argument and a framework. So what are we thinking in terms of creating the data ecosystem that is going to really be released? I hope by 2020, all of this work is going to be complete. So what we are trying to do is that we have this core data frame where we have electronic health records and germline genetics. What else are we trying to actually add to it? So let me spend some time to talk about that. And this is the box which we already have. And then with the consent to link other resources of data, we are trying to link other internal data resources, which are in Michigan but not currently linked. And then what we are trying to purchase some data resources, in particular, SureScript prescription claims. This work is almost complete. And then connect it with state and national death records so that we have good ideas in terms of survival. And so these are the two places where work is being done. This is where something interesting is happening in terms of connecting outpatient EMR as well as EMR from other places where you have been to, right? So how to do that? If there is a patient-mediated portal where you can consent that you can have if I give the data that you can use my electronic health records from everywhere in the world, then I think that's a mechanism which has become popular in terms of linking these databases. And then the other thing from an environmental exposure point of view and a social exposure point of view is that we have geocoded locations for most of these patients. And we're trying to connect to census level data as well as air quality and use ecological aggregate level data in counties and national and state level data in order to create these data frames. So this is sort of the vision, and this is very much a work in progress. Everything sort of seemed to me that it's a fantasy, but over the last 7 years, this has happened. So in the next 7 years, a lot of things are going to happen. So if you're a Bayesian, that's your prior. So what we are launching, and I'm very excited to share, is actually an epidemiology questionnaire data layered on top of the electronic health records data. And so this process started that if we have the electronic health records data, which is imperfect in many senses, and we have the genetics, if we can get good survey data, which UK Biobank actually has, then MGI will be positioned to perform in large meta-analyses, as well as you'll have very well-curated lifestyle and social and occupational and other kinds of data. So what we started, it has been a humongous process, reviewing all biobank surveys, and Maxwell Salvatore in my lab has done a lion's share of this work, evaluating all of the questions and the survey data that people are asking in large biobanks across the world. And in the end, what we have is a baseline questionnaire which actually talks about different phenotypes, different lifestyle choices, different behavioral risk factors. And then the idea is that the different cohorts that I showed you, that the cancer cohort or the mental health cohort or the pain cohort, they are going to have their own baseline surveys. But this data frame, this is sort of a T-shaped data frame, but we have multiple multiple stands to this T, where there is this uniform data which everybody can use, and then investigators do not have to collect this data. It's a kitchen sink of confounders that you can potentially adjust in an electronic health records analysis, but then people are going to ask their questions in a deep dive into the disease focus area of their interest. And then there are some additional modules, which will make the survey very long. The baseline survey is very long. So we have additional modules, which is well-curated questionnaires from the different domains that people are usually interested in collecting data. The idea is that if you're collecting this data, you're going to use the uniform set of questions so that other people can use it too. So this is the data sharing model. So if you and the participants do not have to fill all of them, they have a choice, and we are using a very modern version of an app in order to engage the patient community so that you can do the surveys over a period of time. And also trying to think, we're actually doing a randomized trial about what type of incentive works for different kinds of people, because some people would like to have some information back from other people, like how do I sleep with respect to others? What is my ancestry information? Some people might like a $10 gift card. So we are trying to understand, and this has been an interesting process for myself, and I want to say that we are calling this epidemiology questionnaire APQ or EPIC in some other way, but it is sort of to match with the electronic health records notion that EPIC is the system in which most of the electronic health records in Michigan is actually stored. So Vision 2020. And so this is extremely exciting because you're building a resource which is going to really outlive all of us and going to be used by researchers for generations to come. You are creating a rich data set with a large number of variables and a large number of observations. So in statistics, we'll call that large N, large pre-problem. And this data set will actually allow investigators, because it is so rich and it is not created from a hypothesis-driven point of view, to query this data set from their different angles. And it will be, people will be able to contribute to biobank meta-analysis because we designed the questionnaires in such a way so that there are common items across biobanks. And this is sort of an international worldwide view of sharing data so that we can find people who are more and more similar in terms of a fine, high-dimensional covariate space. They're similar, not just in one or two constructs. So I'm very excited about that, creating this shared resource for the institution and this initiative. Before I move on to the next part, I'd like to pause here and see if there are any questions, because the next part is going to be how I try to use this data resource in order to do some answer some of the more questions in cancer and genetics. Yes, please.
Thank you so much. Is it difficult to collect lifestyle data at the same time as physical samples? And do you know what all of us is doing as part of that course?
Yes, so that's an excellent question. So that's why I did not put this timeline of this survey. We are not doing it at baseline. So what we are doing is an online invitation by email, as well as ask them to download an app. And we are giving them a 1-month period. So we are actually inviting them to participate in the survey 3 months into the study. And then for the retrospective already existing sample, this is the randomized pilot we are doing, that people who are alive and what is the response rate, what works best, whether it's a mailed consent. So it's not at the time, it's not possible. What UK Biobank did was to have them come in and fill out this survey to a center. So we obviously do not have those resources to do that. And I think all of us is also following that pattern in terms of survey and asking them to come and fill it in. Yes, they're not using, because if you think about like enriching, so there are downsides of it. If I do it online, so for Michigan medicine, it's going to work because 80% people actually have an email address. For underrepresented minority and many other cohorts, probably that's not going to work as a model. So you have to have some other means to get them to do this. So in UK Biobank, they had them come in. I don't know how they got that done. UK is a different environment, I guess. So yes, please.
Thank you very much for this. I have 2 questions.
Yes.
Is this data available for public? Can you download them? And the second question, what
So let me go back to this. Actually, let me go back to this slide about the data resources. So right now, what is linked and what it can be easily asked from the data warehouse, the research data warehouse, is actually the clinical data, the electronic health records, and germline genetics, just genotype sequence data. We do not have, so I said that there are some pockets, right? There is a potential to connect with tumor registry and other type of imaging data. So what is happening is that, for example, Laura can talk about her own experiences, that we have this data with germline genetics and electronic health records, and there are investigators who have collected, say, metabolomics data or proteomics data or transcriptomics data. Then they can link this, and so the other pieces are coming from them, but we don't have a large-scale tumor sequencing, for example. We don't have that data for all cancer patients. We do not. We do not. So right now it's germline genetics and electronic health records. Lars, do you know of other omics data being collected?
There's a mechanism in place that people can share their data with the community, but I don't know what the current status is.
So that will be patchy, right? Like the different types of data, epigenomics data, so that would be patchy. But what is happening is that the other way around, that if you have a data set with epigenomics and you need the genetics and the electronic health records, you're just linking your cohort with this bigger cohort to get a more holistic view of the cohort. Yes. So we can point you to the place where you can fill out a form and ask for. It's Michigan investigators, though, at this point, right? It's Michigan investigators. Mm-hmm. Yes.
So other projects like the TCGA, they have what's called controlled access data.
Yes.
You can't access the germline variants for patients unless you go through an EBGAP application
Yes.
process.
Yes.
Can you talk a little bit about the steps that are being taken in MGI to protect patient privacy, or how that's, what's being done so that people's names aren't directly associated with their variants?
Yes. So I'm definitely not the expert on this. Is there anybody from the data office here today? Because someone was supposed to come. But Lars, would you like to comment on that? Erin Kaliba, I think, is doing a lot on that. And in all our analysis, it's a really encrypted ID procedure where we cannot link, like we cannot do the crosswalk. There's an honest broker service at the research data warehouse in order to link any domain of data. And I do think that the university and the institution and Sachin Khetrapal, they are very proactive in terms of protecting the privacy and confidentiality, but I'm not sure of the details. So I'm sorry, but when we get the data for analysis, I know that it is really, really secure. It's very hard without connecting people from the research data warehouse to connect to, we get an encrypted set of IDs, and it's very hard to merge the data of different domains in order to be very cautious about that. Yes. Yes. Do you have any additional comments on this?
So yeah, the data is encrypted. So it's people have to go through an IRB application and data agreement process and a review, and then they get the encrypted data set. But in the future, I think there will be a system in place where people can work on the data in a virtual, secure environment. So they can not download the data, but actually use provided computational resources to work on the data.
Yes. So NHANES and several other national studies actually have this model that you have to go into their workspace in order to access the genetic data, because they don't want to have copies floating around, as to say. So I think MGI is very cautious about that. And so far, the Michigan investigators have a very rigorous process of getting the data. But soon, I think the copies will be limited, and there will be a place where the central, where you have to access the data. But again, my answer will be incomplete. I'm sorry. Yeah. Yes, please.
It's a really wonderful data set. I'm curious, some other cohorts oftentimes have a registration process for the study you wish to do, so that you can be alerted if someone else is doing a similar type of study to be able to collaborate, or to
Yeah.
see who's doing what, et cetera. Do you have any plans for that sort of approach to your cohort?
Yes. So right now, actually, so so this is actually takes to a philosophical issue of how data should be shared and prioritized, because many times in many of the large consortium, what I have experienced is that people claim a concept and an idea and then sit on that forever and don't do it, and it's blocked from other people to look at. So UK Biobank, for example, you can get the data, you can do anything. There is no concept proposal type. For some, the more detailed level of data there is, but there is no concept proposal concept. So I think when Gonzalo, Sachin, and Chad started this, they did not want that proprietary structure so that one can. So what we do is that there is a governance committee where we actually review, see all the ongoing proposals and the IRBs that are going on with MGI. It's so sort of, there is a philosophical thing that whether two people should work on the same thing. So what it has been that people can posit the same concepts, but then when we look at it, we try to connect them together. But I think we have tried to carefully not go into the route of claiming something and not doing it. So you can see, manage all of the different proposals, and then it's a more collaborative spirit in that sense. It's a very progressive, democratic, like many people are looking at maybe the same problem, and in the end, if we all collaborate, we get it to a more accurate answer. So, other questions? All right. So I'm pretty sure I'm not going to get to part 3. That's okay. So Vision 2020. As I said, that this is Vision 2020 is as far as and as best one can see. So I foresee this, that this is a wonderful in-house data resource for people to use. So I want to give you a snippet of an example of a couple of papers that came out recently using this data set, and both are actually led by Lars. I was just helping him. There is an interdisciplinary team of people, and any kind of good science actually really needs a lot of people. Nobody can do it. When I was going through tenure, people asked for like single authored papers that nobody else is on. And now you look at single authored papers, I said, oh my God, is this person doing science? The culture the landscape has actually completely changed. So I wanted to give you a sense of the bias sampling mechanism of MGI and still how we have been able to use this data for some meaningful research. So one thing which was astonishing to me, because I'm sort of new to this land, is that 80%, 78 to 80% of people who you approach actually consent to using their data for research. Every kind of, this is tremendous trust in people and patients in terms of using their data, link their data through social security number, through identifiers to so many attributes, but they just want human health to improve. So this was very inspiring for me. And then, so as I said, that it takes, so there is a backlog in terms of genotyping the data and integrating that with the electronic health records. And we are continuously in an evolving process of reducing that time span. So one thing I want to point out that why I got into the data is actually when I was appointed as the Associate Director for Cancer Control and Population Sciences at the Rogel Cancer Center, I noticed that this data set actually had about 50% patients constantly with a diagnosis of neoplasm. And lo and behold, this cannot be a population sample, right? Population-based sample. So what is the reason? The reason is that the large bulk of the cohort is being recruited from surgery, and cancer patients go through surgery more often. And if they need to have a surgery, instead of going to a local care, they tend to come to a comprehensive cancer center, even if it's for other reasons. So that leads to this enrichment. We did not sample for cancer, but because of the surgical setting, it became enriched for cancer. And maybe it's enriched for other diseases as well. So you'll see that this cohort is enriched for cancer. The mental health cohort obviously is enriched for mental health patients, and then the MEND cohort is enriched for patients with endocrine diseases. Then how do we synthesize all of this data together? So that's something that we all need to think about together. So this is a biased sample. There is no doubt about that. If somebody said this is a population-based sample, they're wrong. So now how am I going to use this for meaningful population-based inference? And initially, I thought that this is really not possible because I don't know why someone is in the sample. I can deal with bias sampling, enriched sampling, as long as I know what is the probability, a certain mechanism of someone being in the sample. But this is like an open game. So just to give you a sense of how the sample, the cohort looks like, and it is, so you can see that the mean age, and it has tremendous wealth of longitudinal data. So median number, so it is not a one-time surgery patient that you're seeing. Most of the patients are in the health system for a long time. And just to give some summary of the richness of the longitudinal data is that median number of visits per participant is 27. And then the length of follow-up is about 5 years, and the number of disease codes. So this is something that you're going to see in this talk. The remaining 15 minutes is of this talk. International classification of disease code distinct is median number is 31, and if it's, of course, a very skewed distribution. So what do we do? Our analysis actually just used 2 domains of data, which is the electronic health records and the genotype data. And I'm not going to get into the process of getting to the ICD-9 to computable medical phenotype. That's a field in itself of medical informatics, but I'm going to talk about something simple. And then how the genotype data was processed. These were standard GWAS type processes in terms of using principal components for controlling for population substructure, genotype imputation. So I'm not going to talk about that either. I'm going to talk about, instead of the genome, I'm going to talk to you about phenome and this phenome-wide association studies and how do we define a medical phenome from this data. So I'm going to try to sort of define an epidemiologic construct. So the first part of my talk was infrastructure. The second part is primarily epidemiology. And the third part, which I'll not get to, is pure biostatistics. So the phenome part, I'm thinking, so when you have these diagnoses, these ICD-9 codes, what you do, what we did in terms of conducting a FEWA study is that, say somebody has a colorectal cancer, diagnosis 153, that person, anytime during the electronic health records, regardless of timing, at least once, and you can rigorize this, saying at least twice or something for phenotyping approaches. You can use clinical notes, you can do better phenotyping, and again, I'm not going to talk about that, to define these are the cases. And then from the remaining people, and say these are 2,000 people in your 40,000 cohort, and then from the remaining 38,000, you choose controls. And you choose controls for colorectal cancer following certain guidelines. For example, people with other cancers or other GI cancers, other neoplasms, and certain other GI diseases cannot be controls for colorectal cancer. So maybe you have 2,000 cases, and then you have 38,000 other disease-free people, but maybe 30,000 of them can be eligible controls. And so you do select controls from the remaining people who do not have that diagnosis, right? And you repeat this process for each of the diagnoses throughout that you see, right? And so you create tons and tons of fake, pseudo, synthetic case control studies. So this is the first processing step for a phenome-wide association study. This was first proposed by Josh Denny at Vanderbilt in 2010, 2013 in highly cited papers. So we are losing a lot. We are losing completely the temporal resolution of the data because we are reducing millions of data points to 1,653 phenotypes, which have at least 50 cases in the MGI databases. This is our pre-processing step. Is this the best way to do this, curate this data? No. And I had lots of issues with that until I'll show you the results, that there is some method in this madness. So, and some people have this, they have been in the electronic health record for 5 years, and some people have been in the health system for few months. And their data are, their phenotype data are actually being treated with equal weight, so which is another statistical issue. So this is the simple analytic pipeline of a phenome-wide association studies, that once you have defined those 1,653 case control studies, you actually group them. So on this axis, as opposed to the chromosomes, you have the disease groups. For example, that you see the circulatory system diseases. I'm interested in neoplasms that are here. and I'm going to run 1,653 logistic regression models. If I wanted to study a particular SNP, I'm going to study, run 1,653 logistic regression models and test for whether the gene is actually theta G is equal to 0, or that there is any effect of the gene or not. And then I'm going to do a Bonferroni correction. And you can see the curse of multiple testing is not so huge as G was, because you have 1,600 phenotypes, not millions of tests that you're running. So you can look at this visualization tool where all of this analysis is actually publicly available, so people can explore this data to generate hypotheses. And so you'll see a lot of plots where for each of the 1,650 tests, I'm going to plot minus log 10 of the p-value, and any point beyond this threshold is going to be noteworthy for follow-up. Any questions about that? How we are processing the data? All right. So what we did was actually do a phenome-wide association study where that G that I showed you in the previous logistic regression model is a polygenic risk score. And many of you have heard about polygenic risk score, so I want to have a show of hand. How many people have heard the word polygenic risk score in this audience? Okay. So everyone, and this has become really a very popular construct, very highly talked about in scientific research these days. And what we did, so polygenic risk scores are some kind of a linear weighted combination of genes which are associated with a particular disease. And the weights are coming from external sources. So I cannot get these weights from MGI. That will be very much overfitting to the data. I can get them from publicly available data sources to create polygenic risk scores. And the data sources that we used is the catalog, the NHGRI, EBI, GWAS catalog, and also several other sources. And I'm not going to get into that. And what people have shown is that if you calculate the polygenic risk score, even if the individual SNPs or GIs have very tiny bit of magnitude of contribution, the polygenic risk score overall have a stronger contribution, the ensemble of small, tiny effects. And so people have also shown that the distribution, so this is sort of a toy cartoon, that the polygenic risk score can discriminate between cases and controls. So this is the distribution of the polygenic risk score in the cases and the control, and you can see that right shift in the cases. So this has been used so there's no clinical guidelines at this point how to use this polygenic risk score for patient communication and patient prioritization and screening. That's something I think is going to evolve, and one of the most exciting stories probably in the next 5 years. And then you associate that polygenic risk score with diseases across the phenome. So for example, here I put a plot. Exactly now I have data. Now I actually have the 1,653 points laid out on the plot. And I have taken the polygenic risk score for prostate cancer and associated that with other diseases. And this is exactly what we cannot get from a case control study of prostate cancer, that what other diseases are associated with this polygenic risk score. That's having access to the electronic health record data. So what I'm going to show you is actually something that stemmed my third part of my research, but this we looked at 16 cancers. I'm just going to show you this result and should then end with the data resource that we have created that you can use. And as you can see that in MGI, it's an academic medical center cohort. We have a large number of cancer patients in this cohort that one can use for research. And these are the number of SNPs or the Gs that went into fit into this polygenic risk score, because the performance of a polygenic risk score also depends on that. It depends on how many cases and controls you have, how many SNPs that you have in your polygenic risk score. And you can see that the odds ratios here are quite strong, quite a bit strong compared to the individual SNPs that you're used to seeing, 1.1, 1.2, maximum of 1.5. So the ensemble of SNPs together is doing something here. And then you can see so these are some of the other cancers that we have looked at, and there are some limitations of sample sizes. And what we have done, one thing is that try to democratize this use of data, because we have done so much work in constructing the polygenic risk scores and curating this data. What you can do is to go to this website, this wonderful website, and I just want to show you this resource before I go back. that you can have, so what you can do here is that different phenotype, and you can explore all of these polygenic risk scores and choose the Michigan Genomics Initiative phenome or the UK Biobank phenome, choose different sources of polygenic risk score. You can choose different cancers so that you can explore these plots yourself. And our goal is to expand this resource to all 1,653 and all ICD-9 codes so that people can use polygenic risk scores and the tremendous millions of data points that we have suffered through. Nobody has to endure that suffering. So so I just end with a sort of teaser. So this is a plot of squamous cell carcinoma. This is a particular type of skin cancer. And we calculated the polygenic risk score for that. And we associated that polygenic risk score with everything across the phenome. And going into this analysis, I thought that this plot is going to be much more noisy and will have a lot of things which are just sort of spurious, because this is really garbage data. So, like, that's my prior. I'm going to change my opinion in a moment. But when you see this, you actually see that everything that is associated with the polygenic risk score for squamous cell carcinoma makes sense. It is actually things that we know are risk factors and associated. But many of them, when you see this association plot, you have to be really careful, because these case control studies are not independent. People who have the skin cancer diagnosis are also getting this diagnosis. And many times it's because of intensified screening because of that first diagnosis. And that's why you are getting it. It's not a true shared genetic association or a precursor. But how can you sort of query? How can you make discoveries, right? So this is this disease I'm going to show you, that how can you discover precursors of, like, so actinic keratosis, it's actually a known risk factor for squamous cell carcinoma, but we did not know this. We are sort of seeing this completely driven by data. And what happens is that if I look at where did the electronic timestamp data help me, the timestamp data helps me to understand if something has a common genetic basis with skin cancer, is it happening before or after the skin cancer diagnosis? And if it is happening before, how much before? And so this is sort of the key point that we are trying to pursue here. The point is that if through genetics we can discover this, in this case this is known, but once we run it phenome by phenome, polygenic risk score by polygenic risk score, are we going to find diseases that are happening many months before the actual cancer diagnosis? That's the point of diagnosis. And many people are getting this diagnosis way, way before. So the goal is that, are there a cluster of diseases, cluster of lab values, cluster of configuration of the covariates, the large p-dimensional covariate is happening long, long before, so that I can start screening here. So that's where precision health and precision prevention is all about. And that's sort of the dream that we are pursuing and probably will be achieved in our lifetime and maybe not. But then I thought, okay, testing is fine and you're getting all of this association, but as a statistician, I could bet 5% of my salary, maybe 2, that the point estimates are not going to be same. So the point estimates have bias. Like, how can a biased sample not lead to biased estimation? For genetics, we are just a win-win story, because we have gold standards to which we can compare the MGI estimates. And I'll show you the squamous cell carcinoma. On this axis, we have plotted the log odds ratios, or the betas that we get from MGI. And this is where it is from GWAS catalog. And you can see that they are not too bad. But think about the GWASs. Every case control study, GWAS and GWAS meta-analysis, is millions and millions of dollars. And in MGI, it's a relatively cheaper data resource which allows you to look at these from so many different dimensions. So as an epidemiologic construct, I think this is very powerful to use the electronic health records data. And the correlation between the two estimates is 0.9. So this happens with many, many diseases. I don't want to overpromise that it looks like this for every cancer, but there is something in this data. And so I, as a statistician, really wanted to know why the estimates are not biased, what's happening into the sampling mechanism and these genetic risk factors that will lead and mathematically prove why they are not biased. So that led to part 3, which I do not have time to talk about. Then we showed that probably because these individual SNPs are not related to your selection or opt-in, because when you are opting in, just think about classic confounding. You have to be associated with Y and X in order to be biasing something. So if your sampling is related to both your disease and your gene, only then it will bias things. And I am a patient in MGI because I went for a hand surgery, but I don't know what the value of the single nucleotide polymorphism is on my genome in order to consent. So once that dependence is broken, you can retrieve a lot of the true information because selection is not dependent on what your single nucleotide polymorphism. But is that going to be true for every covariate? Of course not. Insurance access, lifestyle choices, whether you go to the doctor or not, whether you're getting a surgery or not. So this is an interesting question. So part 3 is sort of mainly putting down the mathematical underpinning of why and when we can retrieve information and association meaningfully from electronic health records. So I'm not going to get into that. I want to just say a couple of things. One, and I'm going to end, I promise I'm going to end on the next slide. So I have a lot of thoughts about this, but this promise and peril of big data. This big data is going to solve all our problems with human health. We're doing all of these things. So I was talking to a friend in economics and business. So there's this concept of this Gartner's hype cycle about every scientific discovery. And it sort of goes through this, and I found that very interesting, that everything we do, you have a trigger, and then you have a peak of inflated expectations, and then you have a disillusionment plateau, and then you recalibrate. So this is the slope of enlightenment where you recalibrate, and then you reach the normative state where you actually think that this is realistic. So I feel like with big data and electronic health records, probably we are here. So please be aware of the thing that is coming. But then I promise you that there is something substantive in here. So I am not going to go through part 3, which has the mathematical layout of this. But I want to show that it is interesting. Like, data science is taking over statistics. Data science is taking over computer science. I hear this all the time. And I just want to show you some popular culture data that what are people searching for in Google. So this is plot from Google Trends. And many people ask me, is your department name going to be changed in biostatistics and data science? I said no, because then we'll be the bads. But look at this. So there is obviously in popular culture, people are very, very interested in looking at, like, searching for data scientist jobs. So it has really exploded. This is a plot. They're searching for this more. It's reassuring to see that the blue line is still above the yellow line. No offense to the computer scientists in the audience. But if we actually, and again, this is very biased data. Not everybody searches on Google. Google Trends has led to devastating conclusions, but still, I'm showing you, since we are talking about imperfect data anyway. So I think that, for the students and the postdocs and the trainees in the audience, that whether we should really do statistics or bioinformatics or data science or some other discipline, I think we have to let go of these buzzwords and focus on our quintessential strengths. People have said that statisticians will be out of jobs when operation research came into the field, economics came into the field, computer science came into the field, applied mathematics came into the field, but we're still here. We are here because we provide something very critical to the experimental field in terms of design, in terms of sampling, in terms of selection bias, which I did not get to talk about. But this is very important. And so I think that you have to train yourself you have to train the next generation. So I'm very proud, incredibly proud of the Big Data Summer Institute that our department runs. And if you did not know this is Big Data. So this is the mantra. And this is the B and the D, if that is a takeaway message from this talk. I really want to thank the organizers of this summer learning. Summer is a time when we can actually reflect and spend in some kind of pondering about different issues. And so really thank Laura and Maureen, two outstanding data scientists on campus, for putting this together and for giving me the opportunity to talk to all of you. So thank you very much.
So we'll take a few questions before we have lunch over in Great Lakes.
I'm wondering if you could speak to how quickly the data that we can access as researchers is updated. That seems particularly important when you're looking at these precursor conditions.
Yeah, so the data is updated, I think every 6 months we get the linkage of the electronic health records, because it's so much work to curate that because you have to update all of the things. So every 6 months, and there is a working group led by Bhramar, Nalamathu, and Jenna Weens, that actually, and Sebastian Zollner, which actually works on the genetics and the clinical warehouse, and then every 6 months, I would say. Is that correct? Will that be correct? Yeah. Mm-hmm. Yeah. We try. Sometimes, sometimes the sequencing data does not come on time, and there are like personnel changes, but 6 months is the target. Yes.
Are polygenic risk scores how GWAS and PWAS data ultimately be used for individuals as opposed to on a population basis?
So this is so close to my heart. I don't know whether I should embark on this dialogue or not. I get very passionate about this. So let me just think about my couple of lines that I can say. So one thing is that I do think that if you look at, and I didn't give this part of the talk, that if we look at the statistically, if you look at the area under the curve, Breyer score, all of the prediction metrics for polygenic risk scores, they are not so impressive at a population level. But what they're really good at is actually discriminating the top 1 percentile or the top 5 percentile with the rest. So if we really care about those high-risk individuals, the polygenic risk scores are actually very useful. That's the first thing. But then top 1 percentile of polygenic risk score may be very few people. So even the odds ratio is high. The number of people in that group, maybe the odds ratio is 8, the number of people is less. And so that's why when you average over the population, you do not get that, because most people. So this is the tension between discrimination and stratifying, picking out the high risk individuals. So I think there is tremendous potential there, the first thing. The second thing, which I am very intrigued in the School of Public Health, is that people in Europe have done certain studies where, with the knowledge of polygenic risk score, even if it's not doing anything, the rate of quitting smoking, changing lifestyle behaviors, knowing that my polygenic risk score is high, has been quite impressive. And I was shocked by that why that is. But if this is true, then even if it's a dummy variable, but if it's changing my behavior with that knowledge, that self-knowledge, I'm going to go for it, right? So I think that those are the two things I can go on and on, but I won't. So yes, Lena.
So the transgenerational effect to public health is quite important. In this cohort, in this biobank, how many of the parented children are there? Even maybe grandchildren?
Very interesting question. So we do not, at this point, because of confidentiality issues, we cannot really identify families. So from the genetic data, we, of course, will be able to, who are related individuals, who are unrelated individuals. Another thing is that so far, MGI was 18 and older. So now Big Bird is the pediatric cohort. So you will not get as much, but you'll still get some parent SIB and case SIBs in the study. This is something that people are probably looking at, that what is the best design to understand genetics, if we can identify case SIB and parent SIB. But so far, we have not really identified families in any project. Will that be correct, Lars, to say? We did never try to reconstruct pedigrees because that will take a lot of, like, go back to the privacy and confidentiality issues and require a lot of, yeah, we have not done that. But if you propose a study through IRB that I'm going to look at, that may be a different process altogether. That's a great question. Yes, Maureen.
So first, thank you so much. It was an excellent talk. Although I would have really liked to hear the last.
I am up for it if you are, but I think lunch is waiting.
So my question is, when you were preparing for the synthetic case control studies, you said for each one you had to determine what were similar diseases, and those patients should not be used as controls. How did you go about doing that for over 1,000 different case control studies?
So that's the FEWAS pipeline. I didn't do it, someone did it for us, right? So 2010, 2013, those papers by George Denny, the FEWAS pipeline actually took a lot of time because they created this ontology of diseases working with the medical informatics people as a biostatistician. I don't have that expertise. It's not possible to do it manually. Yeah, so it's not possible to, there is an algorithm, and that's the sort of FEWAS pipeline that we used in order to group the ICDs into phe codes and then the phe codes into the disease blocks that you saw. Yes. So that's why, you build the architecture. That's why one community builds the architecture for phenotype analysis, another community builds the architecture for creating polygenic risk score, and then everybody uses it, and we rapidly accelerate all the analysis that is not possible, humanly possible, to explore just once. So that's what the vibrant part of it, that's the energizing part of it.
Well, if anyone has any remaining questions, feel free to come to lunch and speak with Bhramar there. Let's thank her.
Thank you.